It wasn't. I'll figure it out.
All right. So, ladies and gentlemen, so we have just kind of one final topic, and then we're going to wrap up by bringing a few things together and talking just a little bit more tomorrow about cancer, which is kind of interwoven through a lot of the things that we talked about in this unit. But today, we are kind of finishing the life cycle that we've been working through in this unit. I told you this unit was kind of focused on all of the different processes, cellular processes, that are going on throughout a life cycle. So we started by talking about just the process of mitosis and the cell cycle that's happening in normal things as they grow, organisms as they grow. We talked about meiosis that forms reproductive cells and how they come back together through fertilization, and some variations on that life cycle. And we looked at meiosis in the Sordaria fungus as a variation on that. And when fertilization happens, that forms a cell called a zygote. That's, again, diploid. Today, we're going to come back to the very, very earliest stages of development and look at what happens within the first days and weeks of the development of a new embryo and how this one cell ends up dividing into a multicellular creature. Okay? So an overview kind of some of the topics we're going to talk about as we go through here. The way that this process happens, the way that an embryo knows to divide and the cells begin to know which should turn into a head and a tail and a back and a front, has to do with controlling the parts of the DNA that are turned on and off and how those genes are expressed in those different cells. We talked about that before, that regulation of gene expression. Your DNA is the same in all your cells. But your brain cells and your heart cells and your stomach cells all use that DNA differently. And it's the same thing from the very earliest stages of development. Different parts of your DNA are turned on and off at different points. And that's what controls the process of development and turns different parts of an embryo into different parts of the adult organism. Okay? Study of this has mostly happened through model organisms, and we'll look at an example of that in 1 second. And stem cells are really important in the process of development. Stem cells are probably something that you've heard of. You may know that they are sometimes controversial in research. My goal today is not to get into discussing the controversy at this point in the trimester. We just don't have time to go there. But I want you to have an understanding of what stem cells are. There are actually a couple different types of stem cells, and so I want you to understand the different types and kind of the different roles that they have played in research and which parts of them are more controversial or less controversial. Okay, so this is just kind of the scientific background. And then if you're interested in this, you'll have an opportunity to go more in depth with these topics towards the end of the year once we're past the AP exam, if this is something you're really interested in, okay? So first, just to give you an introduction to why stem cells are so important, we're gonna watch just a real short video clip about an organism called a planaria, which is a little tiny flatworm. They're about that long usually, about 1 inch long or so. And so we're gonna watch just this little 4-minute video clip because it's a really cool introduction to thinking about stem cells and why they're so important. Why is this going to do this again?
Thanks to the stream for supporting.
Okay. The thing that really sucks is I went and played that video again when that happened last time, by 4th hour, it was totally fine. It didn't do that at all, 4th hour. So we're going to try this once more. I know. Here we go.
Oh, that's funny.
Videos just don't like you guys.
Thanks. Take care. It has to be stream.
All right. We're skipping it, which sucks because that one's actually really cool. It's 4 minutes, and that one actually is super fun, and I'm gonna put that in classroom because I actually would love for you to watch that. Obviously, I can't make you watch it. But I'm gonna link this one in classroom. It is actually really cool. Maybe we'll try and watch it tomorrow if it decides to work then. Who knows? Hang on, I just want to change one other thing about how I'm sharing things since I'm here so I can write on the screen. All right, so basically that was a video about a little flatworm where if you cut it in half or in quarters or in 16ths or whatever, it will regrow. Each little individual section will regrow itself into an entire planaria. Not all of them, and not every section. Some of them, like most of them, there's a certain segment of their body that needs to be included. But planaria are unique because they have stem cells kind of scattered throughout their bodies. And as long as they have those stem cells, they can regrow. So like I said, I'll link that in classroom. It is actually really cool. And you can go and watch it sometime. But we will move on. So what we're talking about here is the early process of development from just a single cell to an organism that looks more like what it actually is. Okay, so this involves 3 different processes. Obviously, there has to be a lot of cell division that happens here, right? And we've already talked about how mitosis happens and how that cell cycle is regulated. So that's a really important part of this. The second thing that happens is cell differentiation, which means the cells need to become different types of cells and start specializing. And the third thing is called morphogenesis. So here are some definitions of some of those. Differentiation is how cells become specialized in structure and function. So that's how just the general ball of cells that you start with at the beginning ends up turning into brain cells, heart cells, lung cells, etc. That's differentiation. The physical process that changes the shape is called morphogenesis, because most organisms begin as just literally a ball of cells. Most organisms, I'm not sure I can think of any right now off the top of my head, are not spherical as adult organisms, right? So somewhere they have to change into their adult shape. That's morphogenesis. And we'll just talk about that part a little bit, probably tomorrow. We already talked a little bit about this. The processes of differentiation and morphogenesis happen because of differential gene expression, so turning on and off different parts of the DNA. And that can actually begin with material that's found in an egg cell. And that's an idea that we're going to come back to here in just a couple minutes. So it's time to start some modeling. Those of you at home, I'm going to be doing this, and I'll kind of tip my camera down so that you can see this modeling happen. We are going to start with the first step. Those of you in class, you are going to be making 2 things. So there's one Play-Doh color that you have 2 colors of. So grab that one and take, let me see. You can use like 1.5 chunks of this. I just want to change what's on the screen. I'm recording here for a minute. And one of them, so take, I don't know, like 2/3 of one and just make it into a sphere. Maybe half of one of them. Let's do that. And this should be the color that you've got the most of. If you want to like be really complete about this, you can like make one teeny tiny bit into like a little tiny sperm cell and the other one into a little tiny egg cell and then smush them together. That represents fertilization. And that right there, so people at home, here's my little tiny sperm cell. There, swims into the little tiny egg cell, smash them both together. And this next cell is a zygote. So that right there is my fertilized egg. That is a zygote. Leave that one on your plate so that you can kind of see this progression. The next thing that happens is that cell begins to divide. So make yourself another ball of the same color that's about the same size. And then you're going to divide it in half, and in half again, and in half again, until you have 16 little tiny spheres. They don't need to be perfect. 2, 4. I'm just going to take my extra pieces. Yeah. So mitosis is happening. I will make all of mine spherical in a little bit. And then that divides again. Let's see. 2, 4, 6, 8, 10.
Or wondering how there's a flame just happened.
Oh,
Where's it coming from?
it's a shadow.
I thought it was a shadow. No, I knew where it was coming from. Right? I told you. Well, I knew it was that window. But like, yeah. Oh, I'm not supposed to use the other one.
If you use part of it, it's okay. We'll need a little bit of it for the next 2 parts too. Okay, so then you can make all those little, so mitosis happened a whole bunch of times here. Now I've got 16 little tiny cells. Yep, so I don't know how many divisions that is. 2, 4, 8, 16. So 4 rounds of mitosis. And then all those little cells are still attached to each other. So you end up with kind of this blob of little tiny cells that are all connected. This is kind of the first stage of development that we're going to talk about. So we're going to pause for just a second there until everybody kind of catches up to that one. And I don't know, people at home, I don't know if you can see that super well there. So it's actually made of a whole bunch of little cells all stuck together there.
See, that's what I was trying to do. I know.
Does everybody understand what you're doing right now?
Yes.
All right. I'll go take the plant off the screen so I don't get distracted. It gets these darn sunny afternoons. It gets really bright in here. We're running together. So those of you at home, while people in the classroom are finishing making their first couple developmental stages here, there is a handout that they are going to be kind of filling in as we go through this activity. I posted it in Google Classroom. The handout looks like this. And it's not in Google Classroom in a form that you can edit, but you can pull this up and kind of make a chart like this in your notebook so that you can follow along and kind of take these notes as we go. What we have made so far are those first 2 stages. And those of you in the classroom, as you finish making this, you can start filling this out as well. When you have just a fertilized egg, that's called a zygote, okay? So that picture is really easy. It's just a fertilized egg. If you want to show yourself that it's fertilized, you could label it 2N. It's diploid. It's got a copy of mom's chromosomes from the egg and a copy of dad's chromosomes from the sperm. They have come together. That's a zygote. This next stage that you just made, or maybe are finishing making, is called a morula, and you don't need to memorize the names of these, but it's things you might encounter if you study this kind of stuff later. So a morula, and I don't care if your picture actually has 16 little circles. It's up to you how perfectionistic you want to be about that today. I'm not even counting. But a morula looks something like that. It's just basically a ball of cells that has divided several times. And as we go through the next little bit of the notes, you can continue filling in the information in this column. And Alyssa and Molly, if you want to pull this up on Classroom so you can kind of see the important vocab words and things to pull out of this, you could have that open. Like I said, I attached this document as a PDF on Classroom today. All right. So we have just made a marula. This happens within the first 3 days or so. These timelines aren't super precise, but within the first few days after fertilization, the cell just divides and divides and divides. The cells at this stage are referred to as totipotent cells. Look at that word. So the last half of it, potent, is like the word potential. Okay, anytime you see that, think potential. What do these cells have the potential to become? And the beginning of this word, toti, is like total, right? These have total potential because the cells within this clump will become absolutely everything in this organism, right? They have the potential to become every type of cell necessary to the development of this organism. This definition will be on the next slide too, if you didn't quite write it down, but it says they are able to become everything. So this clump of cells that you have are totipotent stem cells. A stem cell just means a relatively unspecialized cell that can reproduce itself indefinitely. So these cells can divide over and over and over and over and differentiate into cells of multiple types. And like I said, there are different kinds of stem cells, so we'll talk a little more about this definition as we go through. Stem cells are essential to regenerative medicine. That's where they're mainly used in science. When we're studying kind of how to replace damaged or diseased cells and tissues with new ones. So that's what regenerative medicine means, kind of rebuilding or regenerating tissues. And not all types of stem cells are the same. So its potency, like I said, that part means its flexibility to differentiate or how many different types of cells it can become. And the ones we're looking at right now, like I said, are totipotent. They can become everything. Any questions thus far? So a zygote is just the fertilized egg cell. And a marula is a ball of about 16 cells. You can just kind of describe what they are. Like I said, those are not words that I'm going to hold you responsible for at any point. So here's just kind of an illustration of how stem cells work. A stem cell will divide. It'll go through the cell cycle and go through mitosis like any other cell. But when that happens, one of the cells that's produced, one of the daughter cells, retains that stem cell ability to continue dividing. The other one is what's called a precursor cell that can then, in response to different signals, either become a fat cell or a bone cell or a blood cell or whatever. So it has the potential to become multiple different types of cells. So in this very beginning stage, in this marula, how do these really early cells know what type of cell to become? Those signals come from kind of two main places. One of them are called cytoplasmic determinants, and the other one, the other process is called induction. So we're going to talk about both of those real quick. Cytoplasmic determinants are actually molecules that are found in the cytoplasm of an egg cell. You may or may not know an egg cell contains a nucleus and cytoplasm and organelles and all that stuff. A sperm cell is much, much, much smaller. It hardly has any cytoplasm and it hardly has any organelles. So most of the organelles and things that a zygote receives comes from its mother. Within that cytoplasm, just like in the cytoplasm of any of your cells, there are RNA molecules. There are messenger RNA molecules that were made in the nucleus that are floating around. There are all those other types of RNA that we learned about previously. There are proteins that are being made by the ribosomes in that egg cell and other types of molecules. And those substances might not be distributed evenly within that egg cell. Cytoplasmic determinants are some of those molecules that influence early development of an embryo. And the picture here actually makes this a little clearer than just reading about it in words. So in this first picture, this is an egg cell. And like I said, it's got a bunch of molecules in it. Those could be proteins. They could be messenger RNA molecules that are not distributed evenly. They're the little red circles and they're the little green triangles. And you have more red circles on one side and more green triangles on the other side. If that egg is fertilized, you get the zygote down here. And then when that zygote first divides, this is just the very first division, you can already tell the molecules that end up in the cell on the left are not the same as the ones that end up on the right. There's already a difference between these two cells. And if these green triangles and red circles are signaling molecules that then go into the nucleus of those new cells and start turning on and off genes, that's going to influence those two cells to develop differently. This is a simplified version of this process, obviously, but this is part of where those signals come from. Some of them are already in the egg cell when fertilization happens. Questions about that? The other place that signals can come from is from the environment around the cell. Cells nearby will send signals to each other, and that's a process called induction. And those signals will change how the DNA is read in some of the other cells within that morula. And again, there's a picture here that maybe makes a little more sense. So if you look at the picture in the top left and think about the one that we just saw with the red circles and the green triangles, maybe the cells down at the bottom here were the ones that had a lot of that green triangle signaling molecule, and maybe that molecule turned on a gene in these cells that told them to produce this signaling molecule. And so they start producing a bunch of the signaling molecule and releasing it and sending it to the next layer of cells nearby. And those cells receive that signal. This part should look familiar. Here you've got your signal transduction. You've got a signaling molecule that bonds with a receptor protein that starts a signal transduction pathway that ends by turning on and off genes in the nucleus of that cell. So those original molecules that are inside the egg cell can kind of start the signaling that then cause some of the cells in the embryo to send signals to the cells next to them, telling them to turn into something else. So these cells start talking to each other right away, and that helps to start the process of differentiation and starts turning these cells into different types of tissues. All right, time to make our next model. So here you're going to need about half of a new color. So pick one of your new colors. I'm going to switch this over here. Doesn't matter which one, you'll use them both eventually. So you're going to use about half of one of your new colors. This one, what this would be in reality is a hollow sphere of cells. We can't make a hollow sphere very easily, so you can just kind of shape it into kind of a flattish bowl. These cells that you are making out of a new color actually come from the morula. They come from those same original cells. If you want to really make this look realistic, you can take like the end of a pen or something and like poke little circles into it to make it look like it's made of cells, because really this would be made of a whole bunch of cells. The cells have continued to divide, and this is kind of the first example of specialization. If you don't want to poke little holes in it or little circles in it, you don't have to. But so we've got this little bowl of cells. And inside of that, take some of your original color. It can just be a smaller amount. And make a bunch of teeny tiny cells that are going to sit in a little lump down on one side. So it's going to look kind of like the picture at the top of your slide in your notes. And there's not a specific number here. There would be a bunch of them because this is already divided a bunch.
Oh, no, I just got this.
So you should end up with something that looks kind of like this. So this is our third phase. We'll hold that up a little. The light is not, I need to grab my flashlight. That's what I need to do. All right. Oh, that's better. All right. So that third phase, it's really hard to move things the right way. There we go. Is like a little bowl that actually in real life would be a sphere. We're looking at a cross section with a little blob of cells inside of it. Hello. Thank you. And what this is this represents kind of the first differentiation that happens amongst these cells. Hang on, got to turn my flashlight off. The first thing that happens is some of those cells differentiate into cells that will become the placenta for that growing embryo. That's what this bowl of cells represents. The little clump, which in mine are green, are the part that will actually develop into the organism itself. Okay? So at this point, this happens kind of within the first 2 weeks. And this step of development is called a blastula. Here we have two different types of cells. We have the outer cells here. These are called preplacental cells because they will become the placenta. They're not yet. And this little clump of cells in the middle are embryonic stem cells. So these embryonic stem cells at this point are what are called pluripotent. They can become any type of cell within the organism, but they no longer have the ability to become placenta cells. So they can become everything except placenta. So this is the stage of development where people can get embryonic stem cells. And this is one of the types of stem cells that has been used in research. And these are used in research because they have the ability to differentiate into all different types of cells within an adult organism. They can no longer become placental cells, but they still have the ability to become every single cell type within our body. These are self-renewing in vitro, which means if you take out this little clump of cells in the middle and put them in a petri dish, they will still continue to divide and divide and divide indefinitely. They have, if you remember, we talked about telomeres that kind of get shorter every time cells divide. These stem cells can continue to divide indefinitely because they have an enzyme called telomerase that rebuilds those telomeres. So these cells are kind of immortal. They just keep dividing and dividing. These, some people think these have a bigger potential for research because they are pluripotent. But this is the most controversial line of, or this is where all of the stem cell controversy is. This is the type of research that's the most controversial. Because obviously, if you take out that little cluster of cells in the middle, you have taken the entire developing embryo, and it will not develop into an organism. So this is the type of stem cell research that's really, really controversial. There are other sources for stem cells besides these, and we'll talk about that in just a little bit. Yeah. Yeah, some of them, I tried to get the ones that weren't too dried out. Okay. Questions at this point. So embryonic stem cells are pluripotent. It's at this early stage of development within the first couple weeks. They have the ability to become every part of the body, except for the placenta. Yes. You don't need to know that. I don't even know that. Quite a few. Okay. So then about 14 days after fertilization, that clump of embryonic stem cells differentiates into 3 different layers. Watch closely and listen closely as we walk through modeling this last step. I'm going to switch this over so you can watch the video again. Okay, first you can make the placenta again, so use that same color and make the little bowl like we did before. Remember, in real life, this would be a sphere that completely encloses the developing embryo. It's just really hard to make a hollow sphere and see what's inside of it. So, same placenta color as before. I'm using my bright orange again. It doesn't have to be perfect or beautiful. It's just to remind you that it's there. Then, this is the part that gets tricky. This developing embryo inside the placenta now has 3 layers. So what you're going to do is you're going to take a very small section of your original color, like about the size of a pea, and then you're going to take the 2 other colors you haven't used yet. Do you have 2 other colors? 1 other color. I don't care. Make it 3 colors. Needs to be 3 colors. Share with somebody around you if you need to. For some reason, my count is off. You could even make it like mine. I think I'll make green, yellow, green, just so you can tell it. Whatever colors you have. You just want to have 3 layers. And here's the other thing I'll tell you. As soon as we're done modeling this, I'm going to tell you to take it apart, so don't mash the colors together. Okay? Just loosely. So I'm loosely wrapping yellow around the outside of the green. And then, let's see, I have another color here, so I'll make mine all different colors. It'll be prettier for the people watching at home. Then you're going to wrap another color around the outside of that. And then here's the final trick. I'm going to bring around this really fancy tool called a paper clip. This is what we're going for, people. You're going to slice it in half so that you can see the 3. Let me turn on my camera or my flashlight. So this is actually sitting inside the placenta. You have 3 different layers. Green, yellow, pink inside of the placenta there. Or you could just make it look that way in the first place, like we are. That makes it even easier. The original activity instructions said to slice it in half. So if you want a little paper clip slicer.
Ours is a little crumbly, so it's just...
I know. So we're getting a little crumbly. Here, yes, you may visualize. You may just make a 3-layer, like 3 circles that get bigger.
Okay.
Whatever.
No, I need that one. And then we just have another color and then it doesn't.
So if you kept them all and have them all, you should have the zygote, which is the fertilized egg, the morula, which is just a clump of cells, the blastula, which has the clump of developing embryo cells surrounded by what will become the placenta. And then within that placenta, this inner mass of cells differentiates into an outer layer, a middle layer, and a layer in the very, very center. And each of those will become different tissues within your body. And we'll talk about that here in 1 second. So this final stage is called a gastrula, and obviously it's not the final stage of development, but it's the final stage we're talking about today. And it differentiates into those 3 different tissue layers. And again, these are not details that I'm going to test you on, but each of these tissue layers will develop into different types of tissues in an adult. The ectoderm, that outer layer, will develop into the skin and the central nervous system. So your brain, your spinal cord, your nerves, all that kind of stuff. The mesoderm, the middle layer, will turn into your bones, your blood, and your heart. So your skeletal system and your circulatory system all develop out of that middle layer. And the innermost layer becomes your digestive system and your respiratory system. As we talk through the last few slides here and kind of wrap up this stuff for today, those of you in class, if you want to start taking apart your models after you have sketched them, please separate out the colors as best you can and put them back so it's kind of reset for the next class to do this activity. So you can do that as we're kind of going through the next few. At this point in development, these cells still obviously have the ability to turn into different tissue types, but they are now referred to as multipotent. They can turn into multiple cell types, but they can no longer turn into everything. A cell that's in the middle layer, the mesoderm, can only differentiate into bones, heart, or blood. Those cells no longer have the ability to become skin cells. So these are multipotent cells. Okay. So turning genes on and off throughout this process is what leads to this process of differentiation in different cell types, like we've been talking about throughout. Today, we know about this in terms of molecular changes that alter the expression of genes. The first evidence when we're actually studying these cells is you can detect the RNA before the proteins are even made. You can detect the messenger RNA that's made in this process. So as soon as scientists can detect that messenger RNA, they know that this differentiation process is happening. And then eventually they actually start seeing changes in cellular structure as different proteins are made based on that differential gene expression. Adults do still have stem cells. Adult stem cells are really different than those embryonic ones though. We have stem cells that replace some of our tissues as needed. And so you can see we have stem cells in our bone marrow. You've probably heard of bone marrow transplants. The reason that's effective is they're actually transplanting stem cells. And those bone marrow stem cells can produce all the different types of cells that make up your blood. We have stem cells in our liver, in our teeth, in blood vessels. So there are several different places in your body that you still have stem cells. But the difference is that adult stem cells cannot turn into any type of cell. They're much more limited. So embryonic stem cells, if you take those and put them in a petri dish and give them different signals, they can turn into just about anything. But adult stem cells, you can take like bone marrow cells, and they can become several different kinds of cells. They could become white blood cells, red blood cells, all different types of blood cells. But that's all. You cannot take a stem cell out of a bone and have it turn into a brain cell. It does not have that ability anymore, okay? So adult stem cells are also useful in research and medicine. They aren't quite as flexible in terms of what they're able to become, but they are still useful that way. Stem cell therapies have actually been in use for a long time, probably even before people totally understood what they were doing. Things like bone marrow transplants were some of the very first types of stem cell therapies. And like I said, those stem cells are adult stem cells that can regenerate bone and blood, and that's it. One of the most important recent discoveries in terms of stem cells is something called induced pluripotent cells. So scientists have kind of figured out that they can take certain cells and give them genes that turn them back into stem cells that are pluripotent, that have the ability to become several different types of cells. This is actually a really big deal because if this works and if this research continues to go the way that they're hoping it will, this may make it less necessary to use embryonic stem cells for anything. If we can take something, so this is an experiment that was done. They took a stem cell that had turned into a precursor cell that had turned into a skin cell. They inserted some genes from a stem cell and successfully turned this back into what they called an induced pluripotent stem cell. So this cell now has the ability, it's pluripotent, so it now has the ability to turn into many, many different types of cells. So they can kind of take cells out of your own body. So this is a kind of cool idea. They could take stem cells out of your own body, turn them back into stem cells, and then trigger them to become whatever cell type you need. It would be cells from your own body with your own DNA that they're using to regenerate other parts of you. Which if, like I said, if that continues to be successful, is really cool. Who knows whether this will actually end the debate over embryonic stem cells? Like I said, I don't know exactly where this research is today and how much has been done with this and how much can be done with these induced pluripotent stem cells, but it's at least another alternative. Do you guys have any questions today? Okay, so this is where we're stopping for today. Like I said, the point was really just for you to understand the difference between embryonic stem cells and adult stem cells and kind of see the role that those stem cells play in development and tie that into the gene expression stuff that we talked about last week. Your meiosis lab from Friday officially, I think, is due tonight. Honestly, I'm not grading it right away tomorrow. I'm not caught up on grading right now. So if you're not done tonight, it's not the end of the world. But it is the last week of the trimester. So make sure you're getting things done and catching up on anything else that you might have missing. Okay? All right, ladies at home, that is it for today. So you are free to sign off, and we will see you tomorrow. That's why I was looking.